Hi everyone, welcome to S4 Science. Today's our topic is structure of ovum and the related implantation, fertilization and all. We have finished the menstrual cycle, right? Now that means ovulation occurred, the menstrual cycle, everything we have discussed, and we have to discuss that next portion, right? That is the fertilization. How the gamete fusion is taking place, right? And after that the implantation process, right? All these things we have to discuss. Before that, we need a detailed view of the structure of ovum. We have discussed, but still we need a detailed view. So that's why today we are going to discuss about the structure of ovum. Here you can see I have drawn the picture of structure of ovum, right? I will discuss this first, then I will go for each and everything, right? So like sperms, it is also having a nucleus, right? But here nucleus, you can see that it is called germinal vesicle, right? Inside there will be a nucleolike structure that's called the germinal spore, and there will be nucleoplasm, right? That the cytoplasm of ovum, it's called ooplasm, right? And it is having a covering, right? A plasma membrane is there for that ovum also. That plasma membrane here, it is called the vitelline membrane. Vitelline membrane is a plasma membrane here. And covering to this, there will be thick protective covering, which is very important, called zona pellucida, right? So covering of that ovum, which is a very thick covering, right, which is giving the protection to the ovum, right? So that's actually called the zona pellucida. And after that, you can see that there will be another 2-3, it's mostly 2 to 3 layers will be there, right, covering. You can see that, which is called the corona radiata, right? So this is also giving a protection, and it is also controlling the entry and exit of the cells and all, right? So here you can see that each and every part. First part, we are going from outer to inward, right? The corona radiata, right? Corona, आजकल की वो that corona disease नहीं है, corona radiata, that's a layer. Here you can see that there will be 2 to 3 layers of cells will be there, right? What is the function of this corona radiata? It's actually supply protein, right? So it's actually supply protein for the what we say all the cells, total cells, right? So it's actually supply the proteins, what a nourishment, right? You can say that. The second layer you can see here, that is actually the zona pellucida, right? Which is very important layer. It's actually very thick membrane, right? And it is actually outer protective, it acts as an outer protective covering for the ovum, right? And it's actually formed around the ovum itself. It has another important role. It's actually controlling, right, or regulating the interaction between the ovulating egg and all the freely floating sperm, right? The sperm actually, before the fertilization, the fusion, right, how they will interact, the ovum and sperm, right? So this layer, the zona pellucida, will regulate the interaction between the ovulating egg and also freely floating sperm. That also very important, you have to remember. Next layer we have to go inside, then it is the plasma membrane or the vitelline membrane, right? Here you can see that in this plasma membrane, it contains so much of essential glycoprotein, right, which is important for that sperm binding, for the sperm binding and also species specificity, right? So for the species specificity also and for the sperm binding also, it is very important, that glycoprotein. So glycoprotein rich in vitelline membrane or vitelline membrane, right? And here it is also controlling the entry and exit also, important, the plasma membrane of ovum, right? Next structure we have to discuss the cytoplasm of the ovum. We can see that like the same way in every cell there will be cytoplasm, right? Every living cell there will be cytoplasm. Here also the cytoplasm is there, but here the cytoplasm of the ovum, it is called ooplasm, right? So cytoplasm of ovum is called ooplasm, and again it will also supply that nutrients for the embryo, right? So these all layers actually, it will helping for the developing embryo also, right? So now there will be the nucleus or the germinal vesicle. You can see that it is having that, it contains the genetic material, right, of that ovum, right? So that means maternal genetic material will be enclosed in this germinal vesicle or the nucleus. I hope you understand the structure of ovum. So here we are going to discuss about the fertilization and implantation. We have learned gametogenesis, right? Gametogenesis, we have learned about spermatogenesis and oogenesis, right? I hope you remember, right? Where is the ovum, right? And the ovulation time, the ovum will be in the secondary oocyte stage, right? It will complete its meiosis second only at the time of fertilization. That you have to remember. Then only you will understand all the procedure clearly, right? So gametogenesis हो गया हमारा, right? तो अभी gamete बना कर रखा है. Now they have to fuse together to form the zygote, right? To complete the fertilization, right? So that we are going to discuss here, right? Now the fusion of sperm with ovum, right, is called the fertilization, right? So fertilization is the gamete fusion, right, in which the male and female gametes, that is the sperm and ovum, they fuse together to form zygote, a single celled structure, right? Now, what is the site of fertilization? You can see that. The site of fertilization is the ampullary region in the fallopian tubule or the oviduct, right? So you remember, if you remember, you can easily remember that if that fallopian tubule, if you are considering, there will be a infundibulum, that is the funnel-like structure. There will be fimbriae will be there. Here is the ovary. From ovary the ovum will be released, and that will be captured by the finger-like structure. It will be taken into the infundibulum part, which is the funnel-like opening. Then it will be carried into the ampulla, that is a somewhat wider part of that fallopian tubule. Then there will be a narrow part, the isthmus, which will lead to the uterus. This is the parts of fallopian tubule. I hope you remember, right? So here हमारा fertilization किधर होती है? Here, the ampulla region, not in the infundibulum, not in the isthmus region, especially in the ampulla region it is happening, right? कभी-कभी कुछ case में fertilization can occur in any other area, वो कभी-कभी it will not lead implanted properly. It will not implanted properly, then it will lead to some malformation or some abnormality in the pregnancy and all, right? So correctly fertilization होना है ampullary region, right? Now here the sperm has to, now for the fertilization we told ovum idhar se idhar tak aayega, right? Because the fimbriae like structure, and here there will be so many microvilli are there, right? So that's actually facilitate the movement, right? Help for the movement, right? तो ovum को इधर तक लेके जाएगा. अभी sperm को इधर तक आना है, right? The sperm किधर से आएगा? Sperm has to reach in the vaginal area of the female, right? So that will be that at the time of copulation or the sexual intercourse, the sperm will be released in the vagina, right? And that process is called insemination, right? Along with that semen, right? So the nutritive secretions will also be there. So that now semen along with that sperm and nutritive secretion, that's called semen, that will be released in the vagina of a female by penis, right? So that will be taken into the ampullary region, right? So the release of that sperm, right, into the vaginal area, that's called insemination process, right? Now fertilization happens only if the ovum and sperm transported simultaneously, right? That means the ovum released and it will be wait in the fallopian tubule only 2 days, right? In between that time, right, if the sperms come, then there will be a chance of fertilization, right? Otherwise it will be risky. And the sperm can, before that the sperm can come, and it can be, if it will be insemination happen, right, before 1 week, it can wait at least minimum 1 week, it can wait, the sperm can wait, right? In between that time the ovum is released, then also there is a chance of fertilization. Otherwise there will be no chances of fertilization. That means हर एक copulation will not lead to fertilization and pregnancy. That means all copulation, right, all sexual intercourse do not lead to fertilization and pregnancy. That's why we are telling every copulation or every sexual intercourse, the act will not lead to a fertilization and pregnancy, because it must be released. The ovum and sperm must be released, not released actually, it must be transported into the ovum oviduct simultaneously. Then only there is a chance of fertilization. Otherwise it will be difficult. Ovum can wait at least 2 days, and the sperm at least before 1 week it can come. In between that 1 week time, right? So ovulation happening, right? Then there is a chance of fertilization. That's why whenever those couples who need a baby, they will actually consult their gynecologist and all, and they will make sure that it is the time of ovulation they are undergoing the copulation, right? Clear? Now the sperm contact with the zona follicula. Now if that sperm reached, right, in the ampullary region, once again I will tell that pathway. Here you can see that ovary, right? So from the ovary the ovum will be released, that's the ovulation. Once the Graafian follicle, mature Graafian follicle will rupture and the ovum will be released, right? Ovum ko, that will be taken by the finger-like projections, that is actually the fimbriae. उधर से विले की हेल्प से there will be some villi will be there, that will be help that ovum to move to the infundibulum part, right? So that's actually the funnel part, and that will be taken into the ampulla region, right? Now our ovum is here, right? Now what we need the sperm to come here, then only fertilization will happen, right? So the sperms at the time of that now I'll say insemination, the release will be there, right, in the vaginal region, and it will pass. It is who is having highest motility. That's why the sperm must be that much, what will you say, nourished, right? And who is having highest motility in that. There will be millions of sperms, right? So in that, the sperm which is very strong, that means highest motility will be, that will easily pass through the cervical canal, and it will reach the uterus region, and through that uterus region it will go to the narrower part of the oviduct, that is the isthmus part, and again it can reach in the ampulla part. So now you can see that the sperm reached here. Now they will fuse together, the fertilization will occur. I hope you have an idea about the pathway, right? Now once it will fertilize, that means once it will fuse, then what will happen? You can see this is the ovum region. There will be corona radiata, white line membrane, right? Zona pellucida, everything is there, right? So if the sperm is coming, right, here you can see that it doesn't have any centriole, right? So that's why meiotic division is not completing here, right? Second meiosis complete nahi hoti hai, right? So the ovum will be released in the stage of secondary oocyte. Abhi ये second meiotic division complete होने के लिए it needs centrioles from the sperms, right? Because ovum को centrioles नहीं है spindle fiber formation वगैरह करने के लिए, right? So this sperm will reach here and it will penetrate in the zona pellucida region. That means corona radiata, it will enrich and it will penetrate into the zona pellucida and white line membrane, right? Which is actually helping that. You can see this, we have already discussed the structure of sperm. In that there will be a cap-like structure which is called the acrosome part, in which there will be lytic enzymes, hydrolytic enzymes are there. So many hydrolytic enzymes are there that will help for the penetration, easy penetration of the of these membrane of the ovum, right? So you can see the sperm will come in contact with the zona pellucida, right? And it will induce in the some changes in that membrane also, right? Same time it will penetrate the membrane, it will go inside, that time itself it will induce some changes in that membrane. क्यों induce कर रहे हैं? क्योंकि एक sperm अंदर आ गया, right? So now if any other sperm is also, the same time you can see that there will be so many sperms, but who is having that highest capacity, they will enter inside. Once entry हो गया, then that entail membrane में थोड़ा बहुत changes आ जाती है and that will be sealed, right? Why? Otherwise the other sperm can also enter inside. Then there will be a problem, because at a time one ovum can fuse with one sperm. There are exceptions are there, like twins case and all other some cases are there, but normally it will be like that. The one ovum will be fertilized with one sperm, right? So once the entry that occur, it will induce some changes in the corona radiata region and the membrane will be sealed properly so that another sperm cannot enter inside. Right. So here it will induce some changes in the membrane and block the entry of the additional sperms. Now, the secretion of acrosome, we already told that, right? Which will help that enter the sperm to enter and cause second meiotic division, right? इसके अंदर enter करने के लिए that acrosome. Why that acrosome? Because acrosome के अंदर बहुत ज़्यादा hydrolytic enzymes होती है, right? So it will hydrolyze, right, the membranes, different layers of membrane, corona radiata, the zona pellucida and the vitelline membrane, and will go inside the ooplasm and will fuse with the nucleus, the genetic nucleus, right? So that now the secondary oocyte can complete the second meiotic division. तो ovum की secondary oocyte stage में था, वो complete करेगा second meiotic division, then it will be converted into the ootid, right? So that's actually later, means again it will next term it will convert it into the ovum, and now the second polar body, right, which will form only after the fertilization, that will also be formed. I hope this part you remember. If you are not remembering, please go and check the oogenesis video, then you will have a clear idea, right? I have explained everything in that oogenesis video, right? So now you can see that the sperm will fuse with that nucleus of that ovum, and the second meiotic division of ovum will be completed. It will be converted into ootid or the ovum stage, and also the second polar body which will form after the fertilization, that will also be formed, right? Now you can see here the sperm is also haploid and the ovum is also haploid. It contains the maternal, ovum contains the maternal DNA and sperm contains the paternal DNA, right, the genetic material. That will fuse together, N plus N, and it will form the diploid zygote, which is 2N, right. ये haploid था, sperm and the ovum also haploid. Now it will form the diploid zygote, right. I hope the idea clear, the fertilization and how the fertilization is taking place. So we have finished the fertilization, right. Now that the zygote formed, right. Now the zygote has to be implanted in the endometrial lining of the uterus. Then only it can grow further, right. So that we are going to discuss now, right. So here the zygote, which is a single celled structure, it will undergo continuous mitotic division, right. That means if this is a single cell, it will be undergo continuous mitotic division. It will form 2-celled stage, right, and it will be 4, then it will be 8, then it can be 16, right, like that it will go. So these all stages we will name it as blastomeres, right. So here you can see that the zygote undergo continuous mitotic division. That means they will continue dividing, right, and that means we can say that that division is actually called the cleavage, right, and they will start moving towards the isthmus region and into the uterus plant. That means now the zygote is in the ampullary part of the infundibulum, that fallopian tubule, right. So when it start moving towards the uterine wall, that means through the isthmus region, that time they will undergo the cleavage, right. So whenever it will start moving, so जब वो moving धीरे धीरे नीचे isthmus से uterine wall में चली जाती है, that same time itself you can see that they will be start dividing, right, mitotically divide करती है, right. So that means there will be cleavage happening in between. वो ऐसे ही है, first it will be formed 2, then it will be 4, then 8, 16, like daughter cells will be formed, right. All these stages are known as blastomeres, right. ये सारे stages को हम simply blastomeres बुलाते हैं, right. But when it will reach 8 to 16 cell region, then a cell form, then we will give a name called morula. That means these blastomeres, that means this is the zygote, single-celled zygote. Single-celled zygote will divide mitotically and will form 2, then will form 4 daughter cells, then it will be read to 8 to 16 daughter cells, 8 to 16 daughter cells, then it will have a name called morula, right. And this morula again continuously divide and they will form another structure which is called the blastocyst, right. So this is the structure of the blastocyst, right. So here in this blastocyst structure, it is the stage, right, when the embryo, these all are the embryonic form, right. So when it reaches the blastocyst structure, it will be implanted in the endometrium lining of uterus. But before that, there are some changes happening that we have to discuss. You can see here the blastocyst, right. It is having so many blastomeres are there, that daughter cells are there. These daughter cells are arranged into outer layer. You can see that these blastomeres are arranged in an outer layer, which is called a trophoblast, right. तो सारे blastomeres उसमें cells होती है, वो सारे एक trophoblast की, that means एक outer layer जैसा arrange करती है, उसका नाम है trophoblast, right. And you can see inner there will be some cell mass will be there, which is attached to this trophoblast. You can see here इधर एक blastocele is there, the vacant space is there, और बाकी सारा cells you can see that it is arranged into outer layer और inner cell mass होती है. That means जो cell mass ये trophoblast में attached होती है, right. I hope it is clear, right. ये trophoblast की main function है जो inner cell mass को nourish करना, right. So the trophoblast nourish the inner cell mass. Later ये inner cell mass में changes होती है and that will be converted their division, right. So now this is the stage, right. It will be attached to the endometrial lining. That means अभी ये blastocyst stage में, blastocyst stage में क्या क्या convert that changes होती है. There will be trophoblast, you can see that, which is the outer layer of the blastome is arranged in a outer layer form. उसको हम बोलते हैं trophoblast. और उसके अलावा there will be blastocele will be there, that's actually the vacant space, and there will be inner cell mass. ये inner cell mass ये trophoblast में attached होती है, right. And this trophoblast will give nourishment for the growth of the inner cell mass, right. This is the stage, right. That means ये blastocyst stage में that embryo will be attached in the uterine wall, right? So it will be attached to the uterine wall where the endometrial region, right, inside the, which is having very thick, soft, what we say, high supply of blood vessels, granular, soft part, right? Now it is attached, right? So that attachment is actually called the implantation process, right? After you can see that attachment, you can see that the inner cell mass will divide, right? That will be continuously divide and they will be differentiate. That's actually the differentiation will be there. That means they will be differentiated into three different germ layers, right? That we are telling, the outer ectoderm, right, and middle mesoderm and inner endoderm, right? So अंदर की endoderm होती है, middle की mesoderm होती है and outer की ectoderm होती है. We know down that all the organelles and all the parts of the human body or animal body, that's actually developing from the three germ layers, ecto, endo and meso, right? So this inner cell mass will be differentiated into three germ layers and it is actually protected by the trophoblast. Then the continuous growth will occur as an embryo, right? So here you have to understand this, right? So that time we will give that another name for this structure. You can see that we have discussed that the blastomeres is there, then that will be 8 to 16 stage, then it given as name morula. Morula बाद में convert होती है blastocyst में, blastocyst attached होती है implantation, right? After that वो inner cell mass differentiate होकर तीन germ layers बनाती है, then we will give a name for that embryonic stage, that's called the gastrula stage, right? So this is the gastrula stage, right, which is having the three germ layer stage. Embryonic stage, right, जिसको हम बुलाते हैं gastrula, उसमें three germ layer formation हो चुका होगा, ठीक है? Now after the attachment क्या होती है, कौन सी stage में, blastocyst stage, right? So after the attachment ये uterine wall है ना, the endometrial wall, वो continuously divide होगा, right, and they will rapidly divide and पूरा वो blastocyst को cover करेगा. That means ये आपका यूट्रेन वॉल है, इधर वो ये एंडोमेट्रियल लाइनिंग है, राइट? सो दैट ब्लास्टोसिस्ट आकर इधर अटैच कर दिया, सो दिस लाइनिंग विल बी कंटिन्यूसली डिवाइड एंड इसके ऊपर एक कवरिंग करेगा, राइट? सो इट विल रैपिडली डिवाइड एंड कवर द ब्लास्टोसिस्ट, राइट? तो इसलिए वो उसके अंदर की एक इट एम्बेडेड दैट ब्लास्टोसिस्ट इनटू दैट एंडोमेट्रियल वॉल एंड इट विल मेक इट इज अ पार्ट ऑफ दैट एंडोमेट्रियल वॉल. देन धीरे धीरे उधर से द ब्लास्टोसिस्ट that growth will be there, the gastrula stage, and then it will embryonic growth will be there into a fetus, then 9 months of gestation, like that, right? So this stage of that what we say attachment, right, into the uterus, the blastocyst attachment into the uterine wall, the embryonic stage, right, attached to the uterine wall, that process is known as the implantation. So in this video, we have discussed about the fertilization and implantation in detail. I hope you have a clear picture of these two. If any queries, please let me know. Thank you.